All right, everyone, welcome back for another deep dive. You ready to get our hands dirty with some real microbiology?
Absolutely. Always excited to dig into the fascinating world of microbes.
Today, we're cracking open antibiotics. Challenges, mechanisms, opportunities. That's by Walsh and Wensich, a real treasure trove of knowledge.
Oh, definitely a must-read for anyone interested in the fight against those tiny but mighty bacteria.
We're focusing on Chapter 17, all about antibiotic biosynthesis. And let me tell you, this chapter goes beyond the usual suspects, the non-ribosomal peptides and polygatides. We're diving into some truly unique classes of antibiotics.
Yeah, it's fascinating stuff. It really gets into the nitty-gritty of how these essential molecules are built.
Now, I know our audience is full of brilliant medical microbiologists out there, so let's talk about why understanding how these antibiotics are made should be right at the top of their to-do list.
Well, think about it. We're in this constant arms race with bacteria, right? They're evolving resistance faster than we can discover new drugs.
It's a real challenge, keeping up with those crafty microbes.
But by understanding the ins and outs of how these antibiotics are biosynthesized, we gain a real edge.
So it's about turning the tables on those bacteria, using their own tricks against them.
Precisely. We can potentially tweak existing antibiotics, create entirely new ones, or even hijack their own processes to outsmart them.
Now, that's what I'm talking about, turning those microbes against themselves. So, Chapter 17 kicks off with oligosaccharide antibiotics. I'll admit, when I first saw that word, I was a little intimidated.
Yeah, it sounds a bit daunting, but it's really not that bad once you break it down.
Well, the good news is that it all starts with something familiar. Glucose.
Ah, yes. The sugar that fuels life.
Yeah.
But in this case, it's not about energy.
Okay, so if it's not about energy, what's glucose's role in building antibiotics?
It's more like a Lego brick, transformed into these activated UDP sugars.
UDP sugars. Those sound familiar. I remember hearing about them in biochemistry.
They're essential for building many molecules, and in this case, they're the currency for adding sugars to create some pretty impressive antibiotic structures.
Okay, that makes sense. So how are those UDP sugars used to build these powerful antibiotics?
The chapter dives into two major examples, aminoglycosides and the myenomycins.
Aminoglycosides. Now, those ring a bell. They're those cationic antibiotics, right? Meaning they have a positive charge.
Exactly. They're like tiny molecular magnets attracted to the negatively charged RNA of bacteria.
So it's like a magnetic attraction, but with serious consequences for those bacteria.
That's a great way to put it. They disrupt protein synthesis by binding to the bacterial ribosome, effectively shutting down their ability to make those essential proteins.
So how does the cell actually build those positive charges in aminoglycosides?
Well, it all starts with the biosynthesis of this molecule called DOS, or 2-deoxystreptamine. It's a crucial building block for both streptomycin and kanamycin, two well-known aminoglycosides.
DOS. Got it. And how do they go from regular glucose to this DOS molecule? It can't be a simple one-step process, right?
You're right. It's a multi-step dance, starting with glucose 6-phosphate. Through a series of enzymatic reactions, the cell rearranges and modifies that glucose molecule until, voila, you have DOS.
So it's like a molecular makeover, transforming glucose into this essential building block.
Precisely. And from there, it's all about adding those sugar units onto the DOS core. Those UDP sugars we talked about earlier, they come into play here.
So building an antibiotic is like decorating a cake. I'm starting to like this analogy.
I like it too. The book uses kanamycin B as a great example. Enzymes meticulously add those UDP sugars step by step onto that DOS foundation.
Okay, I can picture it. So they're building up the antibiotic sugar by sugar. What else is going on?
Well, there are some clever modifications happening too. The cell can actually add amino groups to those sugars.
Amino groups, those are the nitrogen-containing groups, right? Why is that important?
It all goes back to charge. Adding amino groups boosts the positive charge of the antibiotic, making it even more potent in disrupting bacterial protein synthesis.
Ah, I see. So it's like adding extra magnets to that molecular attraction we were talking about earlier.
Exactly. Now, while we're on the topic of aminoglycosides, let's talk about streptomycin specifically. It has a few unique twists in its biosynthesis.
Oh, okay. So streptomycin likes to do its own thing. What makes it stand out?
Unlike kanamycin, which has a pretty straightforward structure, streptomycin boasts a central ribose sugar right in the middle.
So it's like the rebel of the aminoglycoside family, adding an extra ingredient to the mix.
And to make things even more interesting, it uses both glucose 6P and glucose 1P as starting materials.
So it's not just sticking to one type of glucose. It's like a master chef, using different ingredients to create a unique flavor.
Exactly. And then there's the creation of dihydrostreptose, this unusual sugar that is unique to streptomycin. It's a multi-step process, a real testament to the intricate enzymatic transformations that bacteria can pull off.
Wow, it sounds like streptomycin biosynthesis is a real masterpiece of microbial engineering.
It truly is. And don't forget the self-protection mechanisms. The bacteria that produce these potent antibiotics need to ensure they don't accidentally kill themselves in the process.
Oh, right. It's like having a safety switch to prevent friendly fire. How do they do that?
Well, streptomycin producers have a two-layered protection system. First, they have an export protein that modifies the antibiotic as it's being pumped out of the cell. Then, there's an extracellular phosphatase waiting outside to activate it. Double protection, double the trouble for any unsuspecting bacteria.
Those bacteria are seriously clever. Now, we've covered aminoglycosides in quite some detail, but Chapter 17 doesn't stop there. It introduces us to another fascinating group, the myonomycins. What makes these guys so interesting?
Myonomycins are like the multi-talented athletes of the antibiotic world. They're not just simple sugars. They've got a pentasaccharide, a phosphoglycerate bridge, and a massive C25 isoprenoid chain.
Okay, now that's what I call a molecular masterpiece. I vaguely remember these myonomycins from a previous episode. They inhibit transglycosylases, those enzymes involved in bacterial cell wall synthesis, right?
You got it. They mess with the construction of that essential peptidoglycan layer, leaving bacteria vulnerable. Now, here's where things get really interesting. Unlike most other antibiotics we've talked about, their biosynthesis doesn't start with sugars.
Wait, really? So how does it begin?
It all kicks off with prenylation of phosphoglycerate, and then those sugars are added.
So it's like building a house, but starting with the roof instead of the foundation?
Yeah.
That's a pretty unconventional approach.
Nature loves to surprise us. And just to make things even more intriguing, there's a late-stage addition of a C5N unit, which is pretty unusual.
A C5N unit. What's the deal with that?
Scientists are still unraveling the secrets of this modification, but it holds potential for designing new drugs in the future. It's like nature is giving us clues for how to create the next generation of antibiotics.
So we've explored the world of sugars and their role in building powerful antibiotics. But Chapter 17 takes us beyond those familiar six-membered rings.
That's right. It's time to delve into the realm of isoprenoids, molecules that go far beyond just your basic cholesterol.
Isoprenoids. Okay, now you've got my attention. Tell me more about these intriguing molecules.
They're incredibly diverse, and some of them have serious antibiotic potential.
So you're saying these molecules are more exciting than they might seem at first glance?
Absolutely. To build isoprenoids, cells use these 5-carbon building blocks called IPPs, isopentenyl diphosphate.
IPPs, got it. So where do these IPPs come from?
And here's the fascinating part. There are two main pathways for making these IPPs, the mevalonate pathway, which is what we humans mainly use, and the non-mevalonate pathway, which many bacteria prefer.
Two different pathways, one favored by bacteria, one by us.
You got it. And that difference is key because it gives us a potential target for developing antibiotics that specifically inhibit bacterial isoprenoid biosynthesis without messing with our own cells.
So it's all about exploiting those subtle differences to create more targeted therapies.
Precisely. Now let's talk about how these isoprenoids are actually put together. Remember those head-to-tail chain elongation reactions from organic chemistry?
Oh, those bring back memories. Only vaguely. Something about adding those 5-carbon units together.
That's right. The C1 of one isoprene unit attacks the double bond of another unit, and so on. And playing a starring role in this intricate dance are carbocations.
Carbocations, those positively charged carbon atoms, right? They make things a bit unstable, don't they?
Exactly. They make the isoprenoid molecules super reactive, allowing them to undergo all sorts of interesting rearrangements, leading to a wide diversity of structures.
It's almost as if those carbocations are like choreographing the whole process, creating all these different moves and shapes.
I love that analogy. It perfectly captures the dynamic nature of these reactions. And the book highlights the importance of these carbocation rearrangements in creating everything from simple monoterpenes, like the ones found in essential oils, to more complex antibiotics like potentamycin and plehensin.
Okay, let's talk about those. First, a quick reminder. How do potentamycin and plehensin actually work against bacteria?
They inhibit fatty acid synthesis, which is, of course, essential for bacterial survival.
Yes, that's coming back to me now. Messing with their ability to build those essential fats.
Yeah.
So how does the cell go about building potentamycin and plehensin?
Well, the book describes this incredible multi-step cyclization process that starts from duranol diphosphate. It's like a molecular origami master at work, carefully folding and bending the molecule to create this complex encarine scaffold.
And those carbocation rearrangements we talked about, they're the ones making all these folds and bends possible.
Precisely. Without them, the whole process would fall apart.
The book also mentions oxidative cleavage and a final linkage to amino hydroxybenzoate to complete the platensomycin molecule. It's like adding those finishing touches to a work of art.
Exactly. Now, another group of isoprenoid antibiotics mentioned in the chapter are the pleuramutolins. They work by inhibiting protein synthesis, binding to the ribosome.
Right. Protein synthesis, another crucial target for antibiotics. So how do these pleuramutolins come to be? Are there biosynthetic pathways similar to what we've seen so far?
While the exact biosynthetic pathway for pleuramutolins is still being researched, the book suggests that similar cation-driven cyclization is likely at play. Given the complexity of their structure, it makes sense that nature would rely on these clever carbocation rearrangements to build them.
Okay, so we've journeyed through the intricate world of oligosaccharides and isoprenoids. What's next on our antibiotic biosynthesis adventure?
Hold on tight, because we're about to enter the realm of the truly unique CP antibiotics. Molecules that seem to defy the rules of typical biological chemistry.
CP antibiotics. I don't think I've ever encountered those before.
That's because direct CP bonds are incredibly rare in biology. Most phosphorus-containing molecules in cells have a COP bond, but these rule breakers have a direct bond between carbon and phosphorus.
Why is that such a big deal?
That CP bond is incredibly stable and resistant to hydrolysis.
So they're tough nuts to crack.
Exactly. This makes them powerful antimetabolites. They can sneak into metabolic pathways and disrupt things without being easily broken down.
Fascinating. So who are the key players in this exclusive club of CP antibiotics?
The book shines a spotlight on two stars, phosphomycin and phosphonothracin. Let's start with phosphomycin. It targets mura, an enzyme we discussed way back in Chapter 3.
Mura. That rings a bell. Involved in peptidoglycan biosynthesis, right?
Exactly. Essential for building that bacterial cell wall, and phosphomycin comes in like a wrecking ball, shutting down mura and preventing cell wall synthesis.
So no cell wall, no party for the bacteria.
Precisely. Now, the biosynthesis of phosphomycin is truly unusual. It starts by converting PEP, or phosphoenolpyruvate, to phosphonopyruvate. But here's the catch. That reaction actually favors going backward.
Wait, so how does the cell make any phosphomycin if the reaction wants to go in reverse?
That's where the next enzyme in the pathway steps in. It quickly decarboxylates phosphonopyruvate, removing a carbon dioxide molecule and pulling the reaction forward. It's like a molecular tug of war, and the cell has a brilliant strategy to win.
So it's all about maintaining that delicate balance, ensuring the pathway moves in the right direction.
Precisely. And there's more to this story. The book also highlights these unusual C-methylation and radical-based epoxidation steps in phosphomycin biosynthesis. These are reactions that showcase the remarkable diversity of chemical tools that nature has evolved to create these complex molecules.
It's amazing how nature can bend the rules of chemistry to achieve its goals.
Absolutely. Now, let's move on to phosphonothracin, the other member of this CP antibiotic duo. What sets it apart is its unique CPC bond, meaning the phosphorus atom is directly bonded to two carbon atoms. Plus, it has this ingenious trick up its sleeve to get into bacterial cells.
Oh, what's this sneaky strategy?
It disguises itself as a peptide. Phosphonothracin attaches to a couple of alanine amino acids, forming a tripeptide that can be transported into cells via peptide transporters.
So it's like a Trojan horse sneaking past the cell's defenses.
Exactly. Once inside, the tripeptide is broken down, releasing phosphonothracin to wreak havoc. And the pathway to make phosphonothracin shares some similarities with phosphomycin biosynthesis, including that initial conversion of PEP to phosphonopyruvate. But it also involves additional steps to create that unique CPC linkage.
Wow, these CPP antibiotics are truly in a league of their own. Now, I have a question that's been bugging me. We've been focusing on how understanding biosynthesis can help us develop new antibiotics, but could it also help us use existing antibiotics more effectively?
That's an excellent question, and the answer is a resounding yes. For example, understanding the biosynthesis of an antibiotic can help us determine its optimal dosage and frequency.
I hadn't thought about it that way. How exactly did that work?
Well, by understanding how quickly an antibiotic is synthesized and degraded, we can develop dosing regimens that maintain therapeutic concentrations in the body without giving those bacteria a chance to develop resistance.
So it's about hitting that sweet spot where we kill the bacteria without inadvertently fueling their evolutionary adaptation.
Precisely. Biosynthesis can also shed light on how antibiotics interact with each other.
You mean like those drug combinations that are sometimes used to treat infections?
Exactly. By understanding the biosynthetic pathways of different antibiotics, we can identify combinations that work synergistically, enhancing their effectiveness and potentially reducing the risk of resistance.
So it's not just about throwing antibiotics at the problem. It's about using biosynthesis to develop more targeted and effective treatment strategies.
Absolutely. It's about moving from a brute force approach to a more nuanced and informed way of using these life-saving drugs.
This is incredible. It sounds like biosynthesis isn't just about making new antibiotics, but also about using our existing arsenal more wisely.
Exactly. It's about understanding the enemy, their weapons, and how we can turn those weapons against them.
I'm convinced. Biosynthesis is a game changer in the fight against antibiotic resistance.
I'm glad to hear it. It's a field full of exciting possibilities, one that holds immense promise for the future of medicine.
Speaking of the future, what are some of the key challenges and opportunities you see on the horizon for antibiotic biosynthesis?
That's a great question. I think one of the biggest challenges is the sheer complexity of these biosynthetic pathways. They often involve dozens of enzymes and intricate regulatory networks.
So it's like trying to solve a giant molecular jigsaw puzzle.
Exactly. But thankfully, we have powerful tools at our disposal, like genomics, proteomics, and metabolomics. These tools allow us to study these pathways in unprecedented detail.
So we're getting better at piecing together those molecular puzzles.
Absolutely. And as our understanding grows, so too will our ability to engineer these pathways for our benefit. Another challenge is the limited availability of starting materials. Many of these biosynthetic pathways require specialized precursors that are not readily available.
So it's not just about having the blueprint. You also need the right ingredients.
Exactly. But this is where the field of synthetic biology is making incredible strides. Researchers are developing innovative methods to create these precursors in the lab, opening up exciting possibilities for engineering biosynthetic pathways.
It's like creating a molecular kitchen where we can whip up all sorts of new and exciting antibiotic recipes.
I love that analogy. Now, shifting gears to opportunities, there are several thrilling avenues on the horizon. One is the discovery and characterization of novel biosynthetic pathways from understudied organisms.
So there are still uncharted territories in the world of antibiotic biosynthesis.
Absolutely. We've only just begun to scratch the surface. There are countless microbial species out there that could harbor novel antibiotics and biosynthetic pathways waiting to be unearthed.
It's like a vast, untapped reservoir of molecular treasures just waiting to be discovered.
Precisely. And with advancements in sequencing technology and bioinformatics, we're becoming more adept at identifying these hidden gems.
It's exciting to think about the possibilities that await us in these uncharted microbial landscapes.
Indeed. Another remarkable opportunity lies in harnessing the power of synthetic biology to create entirely new antibiotics. Instead of merely tweaking existing molecules, we can now design and synthesize antibiotics from scratch, using our knowledge of biosynthesis as a guide.
That's like becoming molecular architects, designing and building antibiotics with tailor-made properties and activities.
Exactly. And this could be a game changer in our fight against antibiotic resistance. We could create antibiotics that are specifically tailored to combat particular pathogens, minimizing the risk of resistance development.
So the future of antibiotic discovery isn't just about stumbling upon new molecules, but also about consciously designing and building them.
Precisely. It's about combining the power of nature with the ingenuity of human engineering to usher in a new era of antibiotics.
Well, I'm incredibly excited to see what the future holds for this field. It sounds like antibiotic biosynthesis has the potential to revolutionize the way we fight bacterial infections.
I share your enthusiasm. It's a field brimming with potential, one that ignites my passion.
Now, we've covered an impressive amount of ground today, from the fundamentals of biosynthesis to its pivotal role in combating antibiotic resistance. But before we wrap up this episode, I want to make sure our listeners are well equipped to tackle those tricky exam questions.
Great idea. Let's revisit those key concepts and make sure everything is crystal clear.
Okay, let's start with the big picture. Why is understanding antibiotic biosynthesis so crucial, particularly in the face of rising antibiotic resistance?
It all boils down to this. As bacteria become increasingly resistant to our current arsenal of antibiotics, we desperately need new strategies to discover and develop novel drugs. Biosynthesis provides us with a roadmap, a way to understand how these molecules are constructed, Armed with this knowledge, we can modify existing antibiotics, engineer new ones, or even outsmart those crafty bacteria by hijacking their own processes.
So it's not simply about recognizing these antibiotics, but about delving into the intricate details of their assembly.
Precisely. It's like having the instruction manual for a complex machine. Once you grasp how it functions, you can tinker with it, optimize it, and even create new versions.
Now let's shift our focus to those different classes of antibiotics we've discussed. Starting with oligosaccharides, what are the key takeaways our listeners should remember?
Well, we learned that even those seemingly simple sugars like glucose can be transformed into powerful antibiotics like aminoglycosides and malinomycins. These molecules rely on the formation of activated UDP sugars, those essential building blocks for assembling these intricate structures.
And let's not forget those clever enzymatic modifications that add amino groups and other chemical decorations to those sugar molecules.
Right. These modifications are crucial for fine-tuning the properties of these antibiotics, increasing their potency and targeting them to specific bacterial targets.
And we can't overlook those ingenious self-protection mechanisms that bacteria have evolved to avoid self-destruction.
Absolutely. Those export pumps and modifying enzymes are a testament to the ongoing evolutionary arms race between bacteria.
Now, moving on to isoprenoids. What are some of the key concepts our listeners should keep in mind?
First and foremost, remember that isoprenoids are a vast and incredibly diverse family of molecules, with many members playing essential roles in both bacteria and humans.
And to build these isoprenoids, cells rely on those essential 5-carbon building blocks, the IPPs, or isopenyl diphosphates.
Right. And as we discussed, there are two main pathways for making IPPs. The mevalonate pathway, primarily used by humans, and the non-mevalonate pathway favored by many bacteria. This distinction is crucial for developing antibiotics that specifically target bacterial isoprenoid biosynthesis without harming human cells.
And those carbocation rearrangements, they're like the choreographers of the isoprenoid dance, orchestrating all those twists and turns to create a stunning variety of structures.
Exactly. Those rearrangements are essential for forming cyclic structures, branching points, and other chemical features that contribute to the diversity and complexity of isoprenoid antibiotics.
And finally, let's revisit those rule-breaking CP antibiotics. What makes them so unique and intriguing?
The key lies in that direct CP bond, a rarity in biological systems. This bond is incredibly stable and resistant to hydrolysis, making these molecules potent antimetabolites that can disrupt essential metabolic pathways in bacteria without being easily broken down.
And their biosynthetic pathways are full of surprises, from the initial formation of that unusual CP bond to the subsequent modifications that create those unique structures.
Absolutely. These pathways highlight the remarkable diversity of chemical reactions that nature has evolved to create these complex and powerful molecules.
Okay, that was a fantastic review. I think our listeners are well prepared to tackle those exam questions now.
I agree. But before we move on to our final thoughts, I want to address a question that often comes up when we delve into antibiotic biosynthesis.
Oh, what's that?
Why are some of these biosynthetic pathways so complex? I mean, why does it take dozens of enzymes and multiple steps to create these molecules? Wouldn't it be more efficient for bacteria to just synthesize them in a simpler way?
That's a great question. It does seem like nature is taking the scenic route in some cases.
Well, there are actually several reasons for this apparent complexity. One crucial reason is regulation. Bacteria need to tightly control the production of antibiotics to avoid wasting precious energy and resources. These complex pathways allow for multiple levels of control, ensuring that antibiotics are only produced when absolutely necessary.
So it's like having multiple checkpoints and feedback loops in place to keep everything running smoothly and efficiently.
Precisely. Another reason for this complexity is diversity. These intricate pathways allow for the creation of a vast array of structurally diverse antibiotics, increasing the chances of finding one that effectively targets a specific bacterial foe.
It's like having a molecular toolbox equipped with all sorts of specialized tools for different jobs.
Exactly. And lastly, these complex pathways often involve unusual or difficult chemical reactions. The multiple steps might be necessary to overcome these chemical challenges, allowing bacteria to create molecules that would be impossible to synthesize in a simpler, more straightforward way.
So it's not always about efficiency. It's about achieving the desired outcome, even if it requires a bit more finesse on a longer route.
Precisely. Nature has shown us time and again that sometimes the most remarkable creations arise from taking the long and winding road.
Well, I'm even more amazed by the ingenuity of bacterial biosynthesis now. It's not just about making antibiotics. It's about doing so in a controlled, diverse, and elegant way that can overcome those challenging chemical obstacles.
I'm glad you see it that way. It's a testament to the incredible power of evolution and the remarkable ingenuity of nature.
All right, it's time for our final thoughts on this deep dive into antibiotic biosynthesis. I'll let you take the lead on this one.
We've spent a lot of time talking about new drug discoveries and strategies for using existing antibiotics more effectively. But I want to leave you with a thought that goes beyond the battle between us and microbes.
Okay, I'm intrigued. What do you mean?
Understanding how bacteria make antibiotics, it's not just about fighting disease. It's about understanding life itself, how it adapts and solves problems.
So it's about learning from those bacteria, even though they can be our enemies.
Exactly. Their ingenuity can inspire innovations in so many fields, not just medicine. Think about chemistry, agriculture, even material science.
So it's about seeing the bigger picture, recognizing that this knowledge can help us in unexpected ways.
Precisely. Antibiotic biosynthesis, it's not just a weapon, it's a source of inspiration, a wellspring of ideas that could lead to breakthroughs in all sorts of fields.
I love that perspective. It really highlights the interconnectedness of everything. Now, before we wrap up, I want to make sure our listeners are ready to ace those exams, ready for a few rapid-fire exam-style questions.
Absolutely. Let's put their knowledge to the test.
Okay, question number 1. What's that defining structural feature of aminoglycoside antibiotics that allows them to bind to ribosomal RNA? Remember those electrostatic interactions?
Take a moment, think about those charges.
And the answer is multiple amino groups, which create a positive charge, allowing them to interact with that negatively charged RNA. It's all about those opposite charges attracting.
A classic example of nature using charge interactions to target specific molecules.
Question number 2. Which NDP sugar is the precursor to DTDP dihydrostreptose in the biosynthesis of streptomycin? Think back to those building blocks.
Remember those intricate steps in how streptomycin is assembled?
The answer is DTDP glucose. The cell takes this common sugar and through some amazing enzymatic transformations creates that unique dihydrostreptose sugar.
It really highlights the power of enzymes to transform simple molecules into complex structures.
Question number 3. What's that unusual initial step in the biosynthesis of both phosphomycin and phosphonates? This one might be a bit tricky.
Think about those CP bonds, remember, and how they are formed.
The answer is the conversion of phosphoenolpyruvate, PEP, to phosphoenolpyruvate by the enzyme phosphoenolpyruvate mutase. That's where that direct CP bond is first formed. Key step in creating these unique antibiotics.
A reaction that goes against the typical flow of phosphate chemistry, showing just how creative nature can be.
Question number 4. What's the key structural difference between phosphonate and phosphonate antibiotics? It all comes down to those CP bonds.
Remember those subtle but important differences in the structures?
The answer is that phosphonates have one carbon directly bonded to the phosphorus atom, while phosphonates have 2 carbons directly bonded. A small difference, but it has big consequences for their properties and activities.
It's a great example of how even small changes in molecular structure can have a huge impact on function.
And finally, question number 5. Monomycin biosynthesis is unique. It doesn't follow the usual rules. What is the initial step in this unusual pathway?
Remember, monomycin biosynthesis doesn't start with sugars like many others.
The answer is that it begins with the prenylation of D-phosphoglycerate. This really highlights the incredible diversity of strategies that nature uses to build these complex molecules.
It reminds us that we should never underestimate the creativity of the microbial world.
That concludes our antibiotic biosynthesis quiz. Congratulations on making it through this deep dive. It was quite a journey, but I think we all learned a lot.
I hope so. It's been a real pleasure exploring this fascinating world with you and our listeners.
As we've learned today, understanding antibiotic biosynthesis is about more than just memorizing pathways. It's about appreciating the elegance, the diversity, and the sheer power of nature's solutions.
And it's about using that knowledge to drive innovation, not just in medicine, but in so many other fields. It's about building a healthier future for all of us.
From sugar rings to isoprene chains, nature's defenses are truly remarkable.
Antibiotics rise, offering hope and a fighting chance.
Biosynthesis holds the key to a future free from the threat of disease. Thank you for joining us on this incredible deep dive.
Until next time. Stay curious, keep exploring, and never stop being amazed by the wonders of the microbial world.